Hi everyone, good morning. This is session 9. We'll discuss the pancreas and the action of it. So, the functions already we have discussed the liver. The liver functions involve liver is the largest exocrine gland, produces the bile production. Bile is alkaline in condition and it includes bile salts, bile pigments. Bile salts play a role in emulsification. Bile pigments are produced from the worn out RBC's hemoglobin. Hemoglobin breaks down and forms into the bilirubin, biliverdin. This is an alkaline juice. However, this is the enzymeless but play a role in the emulsification of the fats. Next, it is a detoxifying organ. That is, by various metabolic activities, prussic acid is formed, which is by metabolism, it is detoxified. So, it is a detoxifying organ. Liver is also considered to be the first check post as what we eat and drink, the food and the water. Before entering into the general circulation, it is being what is called first exposed to the liver. The digested food which are absorbed and are stored in the liver, and in the same way they are what exposed before that is the contaminants are present, the fungi are present, fungal, bacterial, these are being first tested in the liver before being releasing into the general circulation. That's what it is the first check post, and it is an advantage that the whatever the food we have taken, it is being tested. So it is the first check post. Then it is released into the general circulation, and it is a hemopoietic production of the blood cells. At the same time, it is the erythroclastic, that is the born out the old RBCs, old blood cells are being destroyed, so erythroclastic. It is the hemopoietic also. In the early embryonic stage, the yolk sac mesoderm is the hemopoietic organ. Later embryonic stage, the liver is the hemopoietic. Next, it produces the blood clotting proteins, prothrombin, fibrinogen, or what produced in the liver itself, and also the anticoagulant heparin is synthesized. It is with the Kupffer cells, which are generally considered to be the macrophages. Basically, the monocytes, that is the blood cells, which are what when enter into the connective tissues which forms into macrophages. So hepatic macrophages are the Kupffer cells. In various parts they enter, and that is osteoclast in the bone, likewise synovial cells in the joint. So they are Kupffer cells, are hepatic macrophages are for the phagocytosis. Next, it is liver is also playing role in the osmoregulation. That is, we have seen the maintenance of the body fluids and salts, where the angiotensinogen is a protein synthesized in the liver. So it converted into the angiotensin 1, then into angiotensin 2, and by the process in the osmoregulatory process, and it stimulated the secretion of aldosterone, which play a role in the absorption of the salts and the water, thereby osmoregulation. And liver is for performing the functions emulsification, heat production, and storage of the food, and it is also for the what is called two cycles are involved in the liver. One is named to be the ornithine cycle, and another is the Cori cycle. However, Cori cycle is a two-way pathway between the liver and muscles. Generally, the ornithine cycle is involved in the liver where first the deamination takes place. Deamination in the sense removal of the amine from the amino acid. Basically, the amine group is detached from the proteins. Amine. Removal of the amine from the proteins is deamination. This amine is added by hydrogen and forms the ammonia. When ammonia combines with the carbon dioxide, results in the urea. So here the amino acid involved is ornithine and enzyme ornithage enzyme is also involved. So it is a cyclical reaction where the deamination then followed by the formation of ammonia, which combines with the CO2 and results in urea. This synthesis of the urea takes place in the liver, which is by ornithine cycle. So it is an excretory product. We may be misguided that urea is an excretory product synthesized in the kidney. No, it is the synthesized in the liver by ornithine cycle. That is the reason why the large amount of the blood vessel carry the large amount of urea is refers to the hepatic veins. The vein that takes away the blood from the liver is a hepatic vein. Ornithine cycle synthesizes a cyclical reactions takes place in the liver for the synthesis of urea, which involve deamination followed by the formation of ammonia combined with the CO2 and results in the urea. Next, Cori cycle. Cori cycle is said to be generally the muscles undergo the contraction and relaxation. They are for the muscular movements. They are for the movement. So lactic acid is formed. The lactic acid that is formed results in a fatigue. 20% of the lactic acid get oxidized, but 80% of the lactic acid is what taken to the liver, and where it undergoes in a cyclical reaction and results in the formation of what is called glycogen, and then it is brought back to the muscle, which is what forms into the glucose. So the synthesis of the glucose in a cyclical reaction between the muscle and the liver is said to be the Cori cycle, which is what also related to the liver. So under anaerobic reactions, what we see, the lactic acid that is not undergo the oxidation is what taken to the liver and forms into the what is called glucose. So where from it is formed? Non-carbohydrate. So it is said to be the gluconeogenesis. As we have seen, the metabolisms, intermediate metabolism takes place in the liver, which involve glycogenesis, glycogenolysis, gluconeogenesis, lipogenesis. These are all what the liver function. Now we move on to the pancreas. Pancreas is considered to be, pancreas is a mixed gland. Mixed gland. What do you mean by mixed gland? Mixed gland in the sense it is both exocrine and endocrine. Exocrine and endocrine. Pancreas is a pink colored the gland situated in the loop of the duodenum, and it opens into the duodenum and releases its the pancreatic juice. Why it is refers to the mixed gland? It is with the two parts. One, the digestive part, which release the digestive enzymes, that is a part of the pancreatic juice. It secretes the pancreatic juice, so it is a digestive, exocrine, and it also synthesizes the hormones. Basically, 1 million number of the cells are present in the pancreas, which includes both the pancreatic juice secreting as well as the endocrine. Generally, the pancreas is a mixed gland which includes the exocrine part which contain the lobes, name it to be the acini. So it is both exocrine and endocrine. Exocrine part is including the acini lobes, which secretes the pancreatic juice, and another part is the islets of Langerhans, an endocrine part. The pancreatic juice secreting acini part is the digestive part or exocrine, and the islets of Langerhans, which is with the four types of the cells: alpha cells, beta cells, delta cells, and F cells or PP cells, pancreatic polypeptide cells, secretes the hormone. So digestive juice secreting is exocrine, hormone secreting is the endocrine. That's what it is, a mixed gland, both exocrine and endocrine. It is pink color and located in the duodenum loop, that is below the stomach it is located. Pancreas, the exocrine part, if we see, it includes the acini lobes which synthesizes the pancreatic juice. This pancreatic juice released into the duodenum by ampulla of water. Through ampulla of water or hepatopancreatic duct, it is released into the duodenum. The case with the pancreas, via the hepatopancreatic duct, the pancreatic juice is released, and directly by accessory duct it is released directly into the duodenum. The acini, which contain the lobules from which the ductules are what originate, which all join together forming the main pancreatic duct. From the exocrine part where acini are present, from which the main pancreatic duct is arises, and this combines with the common bile duct. Earlier we referred at the liver. The combination of the common bile duct with the pancreatic duct, what forms hepatopancreatic duct. This hepatopancreatic duct, which generally the main pancreatic duct is named to be the duct of Werners, main pancreatic duct. Duct of Werners, which combines with that of the common bile duct and the secretes the pancreatic juice into the duodenum by hepatopancreatic duct or the what is called ampulla of water, whose opening into the duodenum is guarded by sphincter of Oddi. The accessory duct, duct of Santorini, accessory duct, duct of Santorini. This is the diagrammatic view we see. This is the combination of this is the what is called a main pancreatic duct, which is also named to be the duct of Werners, which joins with the cystic duct of the gallbladder and forms the ampulla of water and secretes its secretions into the duodenum, whereas directly a accessory duct, which is this is the accessory duct, which directly releases the juice into the into the duodenum, which is a accessory duct or a duct of Santorini. This is how the pancreatic juice releases into the duodenum. It is an alkaline juice. What do you mean by alkaline juice? Whose pH is more than the 7. That is the pancreatic juice pH is 8.4. So it is in terms of the pH value, it is the highest among all the digestive juices. The pancreatic juice, which is whose pH is 8.4, so it is alkaline, and it is with the enzymes. Enzymes of the pancreatic juice refers to, since it is a digestive juice per a day, about 500 to 800 ml of the pancreatic juice is secreted. This includes the enzymes that are trypsinogen, chymotrypsinogen, pancreatic lipase, name it to be the steopsin. Pancreatic lipase is said to be the steopsin. Next, it is with the carboxypeptidase. It is with the nucleases, name it to be the DNAs and RNAs. What we come to know with the pancreatic juice is, the pancreatic juice have the enzymes of protein digestion, carbohydrate digestion, fat digestion, and nucleic acid digestion, which means protein digesting enzymes, nothing but proteases or proteinases it can be. Protein digesting enzymes are trypsin, chymotrypsin, and carboxypeptidase. However, these are present in an inactive form. Trypsin is in a trypsinogen form. Wherever we came across nosen, it is inactive. Trypsinogen, chymotrypsinogen, procarboxypeptidase are inactive form of enzymes in the pancreatic juice. Now the question is, why they are in inactive form? Generally, if they are released in an active form, in a part, our body parts are made up of proteins, carbohydrates. Generally, the organs or body parts or our cells are made up of the chemicals. These are proteins. So the enzyme, if in an active form it is released in the body coelom or else upon the wherever it is secreted, it is going to be act upon the same part, which means autodigestion takes place. If it is, they are released in the active form, they may damage on the cells they are secreted by, which leads to autodigestion. In order to avoid the autodigestion, the enzymes are released in inactive form. That's what pepsinogen we have seen in the stomach, prorenin also in the stomach. Now here the trypsinogen, chymotrypsinogen, procarboxypeptidase are released in a inactive form to avoid the autodigestion of the cells of the body. So now the question, there in the stomach, HCl is an enzyme activator which converts the pepsinogen into pepsin, prorenin into renin. Then how about here? In the case of the pancreatic juice, trypsinogen, chymotrypsinogen, procarboxypeptidase are converted into active. Basically, an enzyme name it to be the enterokinase. Enterokinase is an enzyme of the intestinal juice. What enzyme activator convert the trypsinogen into trypsin, chymotrypsinogen into chymotrypsin. However, these enzymes which are released are initiate the reaction, but later on, what I mean to say is autocatalysis takes place. The trypsinogen converted into active trypsin by enterokinase. Furtherly, the trypsin itself act upon its own inactive form called trypsinogen and convert it into trypsin, which is named to be the autocatalytic reaction. So trypsin is or the same way in the case of the stomach also, pepsin act upon pepsinogen and convert it into the active pepsin. So autocatalysis process, in the sense the action of an enzyme on its own inactive form and converting into active form. So this is autocatalysis, and autocatalytic enzyme in the acidic region is, that is in the stomach, is the pepsin, whereas autocatalytic enzyme in the intestine alkaline condition is the trypsin. So trypsinogen, chymotrypsinogen, procarboxypeptidases are converted into trypsin, chymotrypsin, carboxypeptidase respectively. Then the pancreatic lipase, which is named to be the steapsin. So it is a fat digesting. Trypsin, chymotrypsin, carboxypeptidases are protein digesting. So proteins are digested. Then the lipase, steapsin, pancreatic lipase act upon the fats. Next, the pancreatic amylase, which act upon the carbohydrates. So proteins, fat, carbohydrates. Nucleases, DNAs and RNAs, ribonucleases, DNAs and RNAs act upon the DNA and RNA. So that is nucleases, which means by pancreatic juice, carbohydrates, proteins, fats and nucleic acids are digested by this pancreatic juice by means of trypsin, chymotrypsin, carboxypeptidase and pancreatic lipase, nucleases, DNAs, RNAs and the pancreatic amylase, carbohydrate digesting. Next, the endocrine part. The endocrine part is named to be the islets of Langerhans. Islets of Langerhans. Basically, these islets of Langerhans are the endocrine part. Endocrine means which do not have the duct. Here exocrine part or exocrine gland is duct. By its pancreatic duct, it is releasing its secretions into the duodenum. So duct gland. What about the endo? Endo means internal. That is, they don't have the ducts. Then how to release or how to the functional role, how their secretions are act on a target cell. That is, since they don't have the duct, they release their hormones into the blood. Through blood they are transported. That's what they don't have, the ductless gland. So endocrine part is the islets of Langerhans. The islets of Langerhans are, the endocrine part contain 4 kinds of cells. That is the alpha cells, beta cells, delta cells and the F cells or the pancreatic polypeptide cells. Pancreatic polypeptide cells, 4 types of the cells. In this alpha cells secretes the glucagon. Beta cells secretes insulin. Delta cells secrete somatostatin, and the F cells secretes, pancreatic polypeptide cells secretes the what is called the pancreatic polypeptide hormone. So this is how we see it is an endocrine. However, the details of this and their function will see in the chemical coordination. However, we'll have certain introduction. Alpha cells glucagon. Glucagon is said to be we have seen in the liver, the glycogenesis takes place, glycogenolysis takes place. Glucagon is a hyperglycemic hormone. Hyperglycemic hormone. It is a hyperglycemic hormone. Alpha cells secretes the glucagon. Glucagon what converts the glycogen into glucose. Glucagon converts the glycogen into glucose. So it increases the glucose level. So it is a hyperglycemic hormone. It is also named to be the what is called increasing the glucose level. Hyperglycemic. Hyper means more. So if the glucagon secreted from the alpha cells convert the glycogen into glucose. So it is breaking down of glycogen. So it is a glycogenolysis. That's what we learn with the help of the glucagon in the liver. The glycogen is converted into glucose, glycogenolysis. That's what it is hyperglycemic. Insulin, beta cell secretes the insulin. Insulin is a hypoglycemic. Hypo. What do you mean by hypo? That is low or less. So insulin convert the glucose into glycogen, which is glycogenesis. So glycogenolysis, glycogenesis, both are one of the intermediate metabolism we see in the liver. So with the help of insulin, the glucose is converted to glycogen. So it reduces the glucose level. So it is a hypoglycemic. And its deficiency what results in diabetes mellitus. Diabetes mellitus is what seen one of the hormonal disorders where the glucose levels increase. Why? Insulin function converting glucose into glycogen. So glucose levels are what increasing. That's what diabetes mellitus. Next, delta cell secretes somatostatin. Somatostatin what inhibits the glucagon and insulin action, which means it regulates somatostatin release from the delta cells. One point to remember that somatostatin have the three sources in its secretion. One from the hypothalamus, one from hypothalamus to the what is called from the delta cells of the pancreas. Third is the what is called we have seen in the stomach and intestine, the argentifin cells which secrete. So from the stomach and intestine, argentifin cells secretes the somatostatin. So somatostatin is of three sources. Hypothalamus, generally somatostatin that is secreted from the hypothalamus inhibits the secretion of growth hormone. That's what it is, growth inhibiting hormone which is secreted from the hypothalamus. The second one, delta cells inhibit the secretions of the both the glucagon and insulin, and in the stomach and intestine it is for inhibiting the gastric as well as the digestive juices. So somatostatin. And the fourth one is named to be pancreatic polypeptidic cells or F cells. This is the fourth one, which is inhibiting the digestive juices. So this is how we see alpha, beta, delta and F or pancreatic polypeptide.